Thank you for the introduction. And also I would like to thank the organizers for selecting my abstract to get the chance to be here and show how you can use the liver spheroids for predictive and investigative toxicology. So hepatotoxicity is a big obstacle on the road to drug development. And despite extensive preclinical and clinical testing, many drugs are being withdrawn from the market. Toxicity testing in animal models are endorsed by regulatory authorities. However, they don't always predict human outcome. So therefore, the recent development of microphysiological systems have raised hope that their incorporation in safety assessment will improve the toxicity testing and evaluation. So to evaluate the use of liver spheroids for safety assessment of small molecules, we are working with the 3D InSight liver microtissue produced by InSvero. This is a cellular model consisting of primary hepatocytes co-cultured together with a crude fraction of the non-parenchymal cells, consisting among others of Kupffer cells and liver endothelial cells. And to avoid intra-donor variability, we are using a multi-donor pool of 5 female and 5 male donors of primary hepatocytes. This model has maintained many essential liver functions, such as albumin secretion, bile acid synthesis, and also drug metabolism. And the model also has a long longevity, up to 28 days, which can be seen, for example, by the sustained size over time, and also here in the ATP content over 4 weeks. Previously, these liver spheroids were evaluated for the ability to predict drug-induced liver injury and compared to 2D culture of primary hepatocytes. In this study from 2017, we saw a good correlation of the cytotoxicity in the liver spheroids and the drug-induced liver injury of the compounds, and we also had increased sensitivity twice as large as the sensitivity for the 2D primary hepatocytes. As I mentioned earlier, these liver spheroids has maintained many physiological features of a liver. Among others, we have the phase 1 metabolism. We don't only have the expression of cytochrome P450 enzymes, they are also tested to be active over extended time, and they are also inducible. Moreover, these liver spheroids, they synthesize their own endogenous bile acid, and we also have the expression of bile acid export pumps such as BSEP and MRP2, which you can see in the histological slides here. And we also have an active bile canaliculi secretion, which you can see in this image as the green fluorescent bile acid analog is being taken up by the hepatocytes and then secreted into the bile canaliculi. Moreover, due to the standardized production of the liver spheroids, we have very reproducible experimental results. I wanted to give you an example here of the ATP assay. This is a standard ATP assay where we test 7 concentration of the test compound. Here, as an example, chlorpromazine is our positive reference compound, and we have 3 drug applications over this 7-day treatment period. Based on the readout and the data, we do a dose response curve, and we can then calculate the mean cellular ATP IC50 value. We compile the data of our positive control and the cellular ATP IC50 value over a period of 2 years and 39 different production runs. And as you can see, we have a very reproducible and stable IC50 value for chlorpromazine, which always lies between 5 and 10 micromolar. So with this, we felt quite confident that we have a stable model, and we were interested in evaluating and assessing its use for risk assessment. And for this, we are using the 7-day treatment of small molecule compounds. And we are comparing the cytotoxicity of these compounds to the DILI rank dataset. This dataset consists of 171 FDA-approved drugs. And to add another layer on this safety assessment and another layer of risk assessment, we decided to include the exposure data by adding the total plasma Cmax value to the analysis. So far, we have screened 82 out of these 171 FDA-approved drugs. And this FDA hepatotoxicity annotation consists of 3 different lists. We have the DILI concern listing with no less or most DILI concern. We have a label section of no match, adverse reaction, warning and precaution, box warning or withdrawn. And the last list is severity class. We analyzed then the data and we normalized the ATP IC50 value to the total plasma Cmax. And then we plotted the experimental data against these different label sections. When we looked at the empirical data, we could see that the distribution of the most DILI concerned drugs had a different distribution to the non-DILI concerned drugs. And by adding then a threshold at 90-fold of the Cmax, we could separate the most DILI concerned from the no DILI concerned compounds. So I've divided here the result into 3 different parts based on the different listings. We start off with a label section, and we can see here, but with the increased severity of the labeling, we have an increased rate of positivity. So warning and precaution has 71% of the compounds tested positive based on our RAC score. In the box warning label, 77% of the compounds were considered positive, and within the withdrawn label, 85% of the compounds were considered positive. We can see a similar pattern when we look at the severity class. At the severity class 5, we have 62% of the compounds considered positive based on the RAC score. Severity class 7, 66%, and in severity class 8, 91% of the compound were considered positive. And finally, in the DILI concern category, we detected 80% of the most DILI concern compounds as positive, whereas 84% of the no DILI concern compounds were considered negative, giving us a sensitivity of 80% and specificity of 84%. Interesting here to note is that the more ambiguous category, less DILI concern compounds, had a divided result of 47% positive and 52% negative. So to conclude this first part, we had a correlation between the in vitro cytotoxicity and the in vivo hepatotoxicity of the drugs. There's a good prediction of the hepatotoxic drugs with the cellular ATP alone. However, the milder toxic drugs or hepatotoxic drugs might need some additional assay, for example, more mechanistic studies or transcriptomics to be detected. And this brings me then to the second part of this talk, where I want to show how we can use these liver spheroids also in more investigative studies for drug injury, liver-induced mechanisms. Thanks to the physiological relevance of the model, we are being able to use these liver spheroids in a few different causality assays. We can investigate, for example, reactive oxygens, the role of reactive oxygen species with a glutathione causality assay, immune-mediated toxicity with an inflammation causality assay, or we can look at cholestasis with bile acid causality assays. But today I would like to give you 2 examples and guide you to 2 of these causality assays. But first, a brief introduction on how we are building up this causality assay. Causality means the relationship between cause and effect. And we are basing these assays on the standard ATP assay. And in addition to the individual treatment of the test compound, we also include a co-treatment of the test compound together with a specific modulator of a certain pathway. And if we then see a shift in the dose response curve or the cellular ATP IC50 value, this would indicate causality. So the first example I would like to guide you through today is the cytochrome P450 causality assay, which aims to uncover CYP450 mediated toxicity. Here we are using aflatoxin B1 as a positive control, and aflatoxin B1 was shown in vivo to produce toxic reactive metabolites when they're being processed by CYP450 enzymes. In the full colored bars, you can see the increasing cytotoxicity of aflatoxin B1 with increased test concentration. However, when we treat the liver spheroids with a combination of aflatoxin B1 and a CYP450 inhibitor, ABT1, in the dashed bars you can see that the cytotoxicity is abolished. Therefore, we are able to recapitulate in vitro the findings in vivo about aflatoxin B1. And second, I would like to demonstrate how the liver spheroids can be used to detect inflammation-mediated toxicity. Inflammatory stress can sensitize the liver for a test compound or a drug and therefore decrease the therapeutic window. And here we can use our liver spheroid to detect desensitization of the liver with inflammatory stress. In this example, we have trofloxacin as the positive control. Trofloxacin was shown in vivo to lead to idiosyncratic toxicity due to the stabilization of TNF-alpha. And trofloxacin alone, you can see in this bright green dose response curve, doesn't have a high toxicity. However, if you do a co-treatment of trofloxacin together with lipopolysaccharides, LPS, which is an inflammatory induction agent, we see an increased cytotoxicity in this model. Therefore, we were able to recapitulate the in vivo data and showing the synergetic interplay between trofloxacin and inflammation in our in vitro system. However, here I would also like to stress the importance of selecting the right model for each assay. Our model here is a co-culture of primary hepatocytes together with inflammatory cells such as Kupffer cells. If you, on the other hand, would use a monoculture of only hepatocytes and do the same treatment, there would be no difference between the LPS treated and the non-LPS treated samples. Again, showing the importance of first choosing the right model for each assay and each hypothesis wanted to be tested. So with this, I've come to the end. I hope that I was able today to show you that liver spheroids can be used both for predictive and investigative toxicology. We also have the physiology of the liver spheroids can result in data which correlates well with in vivo outcome of small molecule drugs. Thanks to the standardized production, we ensure reproducible experimental results. And this, together with the scalability and the cost efficiency, makes the liver spheroids good models for in vitro industrial applications. Therefore, the liver spheroids can be supportive in vitro tool for the critical go, no-go decision making in the drug development process. So with this, I would like to thank you all for the attention and just show you, if you are interested in this topic, we have several posters and we have also more talks. And so please approach us if you have any other questions. Thank you.
Thanks, Anna. Do we have any questions?
Hello, thank you very much. I just had a question. So if you're pooling 10 donors together for your primary hepatocytes, and then you're adding immune cells, how do you control for allogeneic reactions?
So we are always performing a quality control in the beginning. We use these big cell lots, and we have many quality control steps in the beginning. So we know that our model is always performing according to our standard criteria. We are not matching our multi-donor pool with the non-parenchymal cells, so this is not a step in our production.
Then I suppose my next question is, how do you differentiate between damage actually caused by your drug of interest versus damage caused by your allogeneic mismatch?
I'm not sure I can answer that question. Sorry, maybe we can discuss it later on.
Sure, thank you.
Yeah.
Thank you. I have a similar question, also relating to this mix of 10 donors. Are you not losing a lot of heterogeneity as well? Like, have you actually compared this to individual patient spheroids, and if the reaction is the same?
We have the option to also use single donor models if that is of an interest. There have also been some publications in the past with single donor compared with multiple donor. However, we find it useful to pool the donors just to decrease the intra-donor variability. Yeah.
Hi, thank you for the talk. I was wondering, you had a specificity around 73 to 80% depending on your ranking. I'm wondering, with those DILI negative compounds that flagged positive, are the mechanistic studies also used to de-risk those compounds? And can we understand if maybe they are not toxic at all?
So first of all, this is the preliminary data, so we don't know how the specificity and sensitivity will be in the end after screening all these compounds. I think it's an interesting point to look into also the mechanism behind toxicity, or to see if there's a certain toxicity which we detect better than others. With the false positive compounds, so far we haven't looked into the reason for them being flagged as positive, but this could be something we do after we have finished analysis of the whole group of compounds.
Hi, thanks. Here.
Oh, there. Yep.
I have a follow-up question to the heterogeneity question. So your readouts are based on so-called bulk information, but are you planning to maybe include information based on clinical data on polymorphisms in transporters or metabolizing enzymes? Because we know, for example, CYP2D6, CYP2C9 are extremely polymorphic in humans. And of course, this relates to so-called personalized toxicity studies,
For this specific study, we are not considering to do this. We are trying to find a more general approach which can be used for the majority of pharmaceutical companies. However, there could be...
Yeah, but with 10 donors, you can really lose this effect, because maybe your all donors have no polymorphism at all. So you study the same effect in each donor, which is not informative for the general population.
Yeah. It's hard to know. We are not screening for that when we select our donors. But again, this is something which can be selected if this is of interest for a specific project. We're trying now to be broad and to represent the bigger majority of the population, but there is no specific criteria which we are looking for when we are selecting the donors.
Thank you, lovely work. I was just curious, taking from that question, what is the, you mentioned the 5 male, 5 female. What is the ancestry or ethnicity of the donors that you have?
It's a mixed population. It's also a mixed age span. It could be anything from, I think the youngest it's 3 years to 60. We also have different races included in this study. Our samples we're trying to select to have a variability in it. Our samples are originating from American population.
Hi, Erin Gallagher, U.S. Army. How often do you change pooled lots?
The lots, how
Yeah.
can we change them? It depends on how many productions we have and how much we sell, but we would say that something between 1 and 2 years we stick with the same lot. Since it is a multi-donor lot, we have a lot of cells. So something between every year, every second year, we are transitioning to a new lot. And in between, we do bridging studies of a certain set of reference compounds, and we also do all these characteristics of the metabolism and the transporters or expression of cells.
To do a follow-on, so the IC50 data you showed, is that just one lot, or is it that several lots over the 2 years?
From the predictivity
Yes.
study, that is from one lot. So all of it's from one lot and is from one assay per compound. Yeah.
Thanks. We'll have one last question.
Yeah, hi, thanks. Great work. So you showed that you can use this tool as a retrospective analysis evaluation tool right now to check for DILI. Any comments and ideas how you would go about doing that prospectively if you don't have the clinical Cmax value, for example? Because you had that middle range of ATP over Cmax, that sort of like was quite variable. How do you envision to pick the right category for a new compound?
I think, so the idea is to show that our model is relevant and can be used to predict the vast majority of the drug-induced liver injury. And therefore, I think you can use the model also in an early stage of drug development and base your IC50 value when you want to select the most prominent candidate to continue your studies with. If there is no plasma Cmax available, I think you look at the IC50 value and you try to maybe rank the compounds which has the best IC50 value. And also always test as broad as possible to get a nice dose response curve. These are our recommendations.
Thanks.
Thank you, Anna.
Thank you.